So our next speaker for this symposium, it's a pleasure for me to introduce Rob Finn from the EBI in Kingston, UK. Is in charge of the magnify group there, and he will give us some very interesting insight about the tools and resources that are building to translate the environmental and microbiomial sequences that data into main output and services that can be taken out of those data. Rob, please.
Thanks very much, Eric, for the introduction. I assume you can hear me and you can see my slides.
Loud and clear.
Perfect. Well, thank you very much for the invitation to present to you this afternoon. And what I'm going to really present to you is not so much a direct sort of research output, but how the research that we do within my team translates to the databases that we also produce within my team. And I'll try and reflect about how one influences the other. And I'll try and give you a view of some of the things we're thinking about and how we make those services available to the community. And when I say the community, this is people like yourselves who may want to undertake microbiome research, but may not have the resources or the pipelines necessarily in place to conduct the research. So as Eric mentioned, I'm based in the UK at the European Bioinformatics Institute, but from the opening slide, you'll see I can try and get Europe on my slide quite a few times, and I consider myself European. So I'm going to just start this off in terms of a quick 101 of microbiome research and where we are today. And really, this field has been evolving rapidly over the last 10 to 15 years. And really, you know, the range in which of microbiomes that are being investigated these days never ceases to amaze me. And so people are investigating the microbiomes in deep ocean abysses, and there are microbiome studies happening on the International Space Station. On this slide, on the left-hand side, I've got some exemplar microbiomes. There are the three classical microbiomes that I would consider that have been sort of established in the field, which is human-associated microbiomes, marine or aquatic environments, and soil. But now, in more recent years, you're seeing many more targeted studies that may be looking at discovering of new enzymes for green chemistry, biofermentation, and even things like oil-filled souring, where actually you get bacterial populations utilizing the complex hydrocarbons. Again, if we cast our minds back a few years, maybe a decade, very much the field was limited to looking at the prokaryotic fraction and using PCR-based methods, typically against the ribosomal RNA, the so-called amplicon approaches. But now people are investigating the whole microbial fraction from viruses, prokaryotic organisms, and single-cellular eukaryotes. Now we're also seeing that there's a shift to conducting shotgun metagenomics, so extraction of the DNA. Metatranscriptomics is also commonplace, but not as much as the whole shotgun metagenomics. Whilst the short read basis approaches are still very dominant, we're beginning to see more and more long read technologies coming in. And I think that is definitely the future for the field. And really what we're trying to do with magnify, the resource I look after, is to try and accommodate for this large and diverse array of different studies associated with microbiomes. So as I mentioned, magnify is a free-to-use resource, and we have three key areas that we try and tackle. The first is the archiving of the sequence data. The second is the assembly of metagenomic and metatranscriptomics data and the analysis of that such that people can then browse that data. Now, the tagline is the browsing of microbiome data. What we really mean there is any sequence-based microbiome data. So that's anything from metabarcoding all the way through up to metagenomics and metatranscriptomics. Now, I'm not going to go through the website, but there's just some illustrations of the sorts of things you can get out. But what I really wanted to do is say that magnify gives you that good entry point to analyzing data and actually comparing different data sets and discovering data. But I really wanted to contrast that service with the things that we're doing in my research group. And so I'm going to switch tack slightly now. So one of the big areas we've been investigating in my research team is the human microbiome, particularly the human gut microbiome. And one of the things that's always fascinated me is the fact that, you know, where you and I are 99% identical genetically, our microbiomes may differ by up to 80 or 90%. And I want no critique about the UK diet associated with that. In our microbiomes, there are thousands of different species of bacteria that have been identified, and there is about 1-to-1 ratio of cells in our microbiomes to the number of cells in the human body. We carry about 2 kilograms of bacteria around with us, and really there's been this constant sort of effort in the culturing world to try and isolate organisms. And also with the advancing techniques of metagenomics is to actually apply computational techniques to actually start getting at the genomes of your microbiome. So how have we done this? So this is very much a development of a pipeline within my research team. And so we start off with the raw reads. We assemble those using metaspades into longer contigs. And then we group those contigs into sets that we think have come from the same organism, a process that we refer to as binning. Then we will do some quality assurance on those. There are lots of different ways we perform quality assurance now. Back when we did this study about 3 years ago, we focused on using a tool called CheckM, which uses single copy marker genes to assess completeness and contamination. As a quick aside, if you have 10 single copy marker genes and you find 9 of those 10, you're considered 90% complete. If you find one of those 9 marker genes twice, that shouldn't happen because they're single copy marker genes, and therefore you're considered to have 10% contamination. But one of the key things that we did in this particular piece of research was actually start looking at how good these metagenomes assembled genomes, once they've been quality assured, how good they are against the best catalogues that we could find for the human gut microbiome. And I was very lucky to work very closely to another lab in the institute right next door to the EBI, and Trevor's Lawley's group had been culturing a lot of bacteria from the gut microbiome using some quite sophisticated culturing techniques. And so we had access to a lot of genomes that weren't in the reference databases, but we could also go to the reference database. So now just taking a slight aside and thinking about, okay, all of this genome generation, and actually how should this be happening in the ideal world and in the service world? At each one of these stages, we actually want to capture the intermediates. And one of the things we've been working with is my colleagues at the European Nucleotide Archive are actually getting systems in place to capture these appropriate parts of the process that we go through. So there's a metagenomic assembly going into ENA, and then this process of capturing genomes and then generating reference collections, and they are going into magnify, and you'll see a little bit of that later. But one of the key things associated with these metagenomes, assembled genomes, is how reproducible are they, and how representative of an actual genome are they? So if you have, the first question is you've got 2 independent groups doing the same analysis using different tools, do you get the same answer? And then if you can find an equivalent isolate genome, how close is that? And then there's the final question is how comprehensive are these questions? How comprehensive are these collections? So I'm not going to go through all of this in minute detail. I'm going to give you the headlines of this story just so that we can keep focusing on some of the different aspects of those questions. So we started with 13,000 human gut metagenomic data sets. We applied that assembly and binning process, and we identified nearly 2,000 species. And then what we actually found out is from comparing against the reference databases, about 70% of those were yet to be cultured. We were not the only group conducting these large-scale analyses. There were two other papers, one from Nicholas Agata's group in Italy, and the other one from Nikos Kapides in the US. And what we actually did is we took all of those data sets and then merged those together to come up with the unified human gut genome catalogue. And that now contains 4,644 representative species, and of which 80% are yet to be cultured. So on the right-hand side of this slide is a phylogenetic tree showing those representative species. And I'll just walk you through this tree. So coming off the tips of the phylogenetic tree are different colors. I want you to just focus your eye on the green, and they are all those that are uncultured. The other colors represent where there's an equivalent isolate, with the blue representing where that isolate has actually come from the gut microbiome. Then around the outside of the phylogenetic tree, we have a circle that's color-coded according to the phylum, and then the whiskers coming off the very outer ring actually indicates the number of times that representative species was actually recovered. What you can see here is that there is an uneven distribution of the number of times that a genome was uncovered, and actually typically the uncultured are tend to be less common. And what we also find is that the cultured genomes happen to occur in very specific areas along this phylogenetic tree, showing that actually what we do know from culturing is somewhat biased. This catalogue is very comprehensive, and actually underneath those near 5,000 species are over 210,000 genomes corresponding to 170 million non-redundant proteins. Now, if you want to put that in context, that's essentially the size of UniProt for proteins and the size of GenBank for genomes. Here is another way of how we actually did all of the clustering, where we took the different metagenomic studies that are shown in green and used various different databases for isolates shown in blue. And when we talk about something being identified with an isolate genome, whether it's a purely isolate cluster or a mixture of metagenome assembled genomes, we still considered that as a cultured representative, and only those clusters that were purely these metagenome assembled genomes would we actually consider that uncultured. We've then done pangenome analysis on this, which is another huge set of data. And really to say that we've got all of this data, and it's a really interesting research project. But then came the question of actually how do we present this? And it is a challenge, and we'll discuss this a little bit more later on. To return to the question about how good are these metagenomes assembled genomes with respect to an isolate, and how reproducible are they between the different experiments, this is what really this slide sums up. So on the left-hand side, it's comparing to an isolate. And what we took was a metagenome assembled genome, and then we aligned that to a reference where a reference could be found. And we consider a good match to an isolate genome as having 60% of alignment between the two genomes and average nucleotide identity of greater than 95%. So in the top right-hand corner are those that satisfy the 60% alignment overlap. And then if we start drilling down and looking at the quality of the alignment in terms of average nucleotide identity, you can see that vast majority of those are are greater than 95% ANI, and even some were greater than 99% ANI, which means essentially you have the equivalent genome. what you'll notice is that if the keen-eyed amongst you will notice that it doesn't quite go along the diagonal, and that's because some of the repetitive regions you'll never assemble out using metagenomic approaches. In terms of reproducibility between different groups, by having these 3 large studies, we could actually start comparing them. And on the right-hand side is an upset plot of the comparison of the MAGs. The first column in that upset plot indicates that actually all 3 groups agreed, and that's actually the largest, and that was quite really quite interesting. The next largest was actually from the Italian group, but when we looked a bit deeper, this was down to actually the fact that the Italian group had access to some hunter-gatherer samples that were not in the public databases at that time, and so were unique to their study, and that accounted for most of those genomes. And then the next 2 columns indicate that actually 2 of the groups agreed on that genome, and that really shows you that there is a big overlap between those 3 studies. So we've now have all of these genomes. We started to ask questions about why have they not been isolated before? And really what we've got here is trying to take different phyla, separating those into cultured versus these metagenome assembled genomes or uncultured. And what you have here are 3 phyla where we've looked at effect plots of different functional groups. And if you're in the positive direction in slides, you're more common in isolate genomes, more common in metagenome assembled genomes, and in the negative direction, you're less common in these metagenome assembled genomes. And so what you can see across those 3 phyla is actually there are a number of functions that are highlighted in bold, so cell redox homeostasis, oxidoreductase complex, antioxidant activity, and peroxidase activity. And they're all functions associated with dealing with oxygen, and they're actually depleted in these MAGs. And one of the things that, where we think that actually we haven't seen these metagenome assembled genomes in culturing experiments is that they seem to be extremely sensitive to oxygen. So returning back to magnify, we are now making available these species representatives along with all of their annotations through the website. You can get access to all 210,000 of those genomes through the FTP site, but you can also start drilling down to the Pangenome analysis. And if you're interested in those, then I would encourage you to go and look at the website. But again, I'm not going to show you those. What I'm going to talk to you about is some of the other things that we've got coming. So we have a large human skin collection and also a chicken cecum microbiome. And again, on these, I'm going to show you phylogenetic trees. They're color-coded in slightly different ways. On the left-hand side, the MAGs are color-coded green coming off the tip. And again, you can see there's a fair amount of novelty there, but there's nowhere near as much novelty from the skin microbiome. And they're also interspersed the cultured genomes with the metagenome assembled genomes. So it's quite a distinctive pattern. With the chicken cecum microbiome, this is looking at how the microbiome influences production. And here on the outer ring, the green is indicating the novelty. And here you're seeing huge amounts of novelty, mainly because the chicken microbiome has just not been investigated anywhere near as much as the human associated. So we have all of these collections coming. I'm also told by my team that our marine collection is also soon going to be available. And so what does this all mean? So here is a slide of looking at the assemblies in the European Nucleotide Archive over time. And the the x-axis indicates the time and y is the number of submissions. The grey area looks at the totality of all assemblies in the European Nucleotide Archive. The blue line represents those that have just come from traditional isolate genomes. And what it actually looks like is that's flat, where we know that's previously been going exponentially. But in context of when we start adding in the environmental assemblies, which is shown by the green lines, the totality of all environmental assemblies is shown by the solid line. Then the dashed lines the longer dashed line indicates those assemblies coming from magnify, where the smaller dashed is from the community. And what you'll see is actually we had to stop in magnify. The reason why that's flat is because we actually were breaking the European nucleotide archive systems for actually the numbers of assemblies that have been going in. And these assemblies have been coming in both in my research group and also for things that where people have requested assembly. So going and just looking at magnify in a slightly broader context, here is a representation of all the different data sets that we have in magnify, and really just focusing on the amplicon and metagenomes and assemblies. And that's what you're seeing radiating out. In the center is a tree representing biomes. So the yellow represents engineered biomes, brown, that brown color, soil, the blues, aquatic environments, green plants, and then animal-associated microbiomes. Right in the center are the numbers of studies, and then coming out of that are in the red color is the number of amplicons, and then further out from there is the metagenomics. And so we now have huge numbers of amplicons. We've got over 350,000 amplicon data sets. But one of the things that's quite intriguing is the fact that we've really switched to driving the number of assemblies we have for metagenome assembled genomes, because you can get to not only the fact that you can get to longer contigs and proteins, then you can start getting access to these metagenomes assembled genomes. Within Magnify, you can request analysis of your own data if you submit it through us into ENA. You can also request analysis of a public data set. So that allows you to if you want to have your data set analyzed and another data set analyzed using exactly the same pipeline and then compare them, this allows you to do this. And there's an option for doing that through the Magnify website. Obviously, if you request a huge amount, you'll have to wait, because we don't have infinite compute resources. So we try and balance this out across different people to make sure that the resource allocation is fair. One of the things that underpins Magnify is a very boring but very important part is the fact that we describe all of our pipelines as workflows, and they are formal descriptions that are described using the common workflow language. What this does, and I'm not going to labor the point too much, and this is really driven through work with Elixir, is the fact that these are reproducible. So we know exactly which tools, which versions of the tools, which reference databases, and which parameters we have used. So, and what this allows us also to do is quickly swap out different databases and try different tools very quickly to see how much those impact informatics analysis. So I'm now turning to what's immediately coming available in Magnify. And so this really comes back to the research group. So having established that bacterial catalog, we were interested in the viral component, particularly the phages. And so we have gone through a process of taking some of the high quality isolate genomes, developed a pipeline called Verify that uses a number of public tools and some in-house profile HMMs for trying to actually identify taxonomy of those phages. Once we've actually identified contigs that we think are phages, we undergo a lot of filtering and quality control, and we've come up with a gut phage database. And then we've gone through the same process, applying that to 28,000 metagenome assembled genomes. And there we have the mere problem now of the fact that we have 142,000 quasi phage species from the human gut microbiome. My research team have caused me headaches because I now have to try and think of ways of exposing this through Magnify, and this is very much work in progress. But what is available or will be in the next version of the pipeline, which should be due out late summer, is the fact that this Verify pipeline will be integrated into Magnify. And we'll actually be going back and reanalyzing all our assemblies, because you apply this to assembled metagenomes, and this is going to lead to a huge number of phage. As part of trying to link phage to genomes, we'll also be running CRISPR-Cas finder to link that host, to use the spacers to actually link the phage to their host. And this is really a representation of the schematic of Verify. We have this unique part that is in the sort of red color on the right-hand side, which is very much about trying to assign taxonomy. And it's a whole series of steps to try to assign that. And it's taken a long time to actually come up with the best solution to try and combine different tools to access as much of the viral component without overcooling. But what's quite clear with the viral component is we just know so little about viral component of microbiomes. One of the things that really illustrates this to you is when we started looking at the results from the human gut phage data collection, what we did is we looked at the prevalence of the different viruses or phage. And what we found is that the crass phage, which is well known to be highly prevalent, was number one, but then there was a unique cluster which we've called the goober phage, that was the next most abundant. And this is nobody had identified this before. And this is particularly prevalent in Europe, but we find it in all samples. Possibly on this slide on the right-hand side, you might notice that North America is down in terms of prevalence. And that's an interesting change in the geographical distribution. But one of the things is that we also find this goober phage in other environments. So more natural environments, not just host associated. So there's an interesting new piece of research going on there. This goober phage is related to the crass phage. So the goober phage is on this branch coming out. And if we start looking at that goober phage, there are two distinct groups, which with group one that can be further subdivided into three other groups, which probably represent species or subspecies. It's a little bit hard to know the exact distinction between those. And finally, just to really summarize this up, we are also worrying about the eukaryotic fraction. This is at the earliest sort of stage of research going into magnify. But Paul and Sarah, both PhD students in my group, have been looking at different aspects of this. Sarah has come to me from Julie Segre's group, where they have a lot of skin microbiomes. This is particularly good if you're looking for the eukaryotes, because it's got a much greater abundance, but it's got much lower biomass, so it causes other problems. And one of the things that we've been doing is trying to overcome the fact that eukaryotic genomes are bigger and have more repetitive regions, and they have more variable composition. And Paul has been trying to address some of the lack of tooling around here. So his tool, I'll come back to that tree in the next slide after this. Paul has developed a tool called EuCC that uses single copy marker genes, a bit like CheckM when I first introduced to that. But the problem with eukaryotic genomes is there's no universal set of marker genes. And so what we actually do is do a double pass when we have what we think is a eukaryotic metagenome assembled genome. So we first of all get an approximate taxonomic classification, then choose clade-specific marker genes. So finally, what you can see in this slide from applying that tool and other approaches to the skin microbiome is on the left-hand side, we have a tree of mags and isolate eukaryotic genomes. The metagenome assembled genomes are in blue and the isolates are in red. The blue genomes, where there's a very short branch length, are where we've actually recovered something that is equivalent. But you'll notice in that list, there are actually three that are novel. So there's one at the top here, one in the middle, and one towards the bottom. And then when we look at the distribution of those genomes on either healthy volunteers, which is, if you look on the right-hand side, is the top portion, or at different body sites, which is the lower portion of that, the first novel genome is really, really abundant on different individuals and across different body sites. The next novel genome is actually more restricted to certain individuals and is at lower abundant across different body sites. Whilst the final genome seems to be very specific to one individual and a handful of body sites. And this is really sort of showing you that we can really that we can really start accessing the whole microbial component from a genomic perspective. And that really leads me to just say thank you to all of these people who have contributed. My team is on the left-hand side. I've mixed up my research team and my service team and all of my collaborators. And thank you for listening, and I'll take any questions. Thank you.
Thanks a lot, Rob, for this excellent talk and this broad overview of what kind of tools you are using, what kind of approaches, and the large collection of environments you're looking at. That's a tedious work. It's I think that there are a lot of questions. The first one from Etienne is about the tools and the way that you are doing the taxonomic assignations on mags, and that's from bacterial or archaeal mags. Can you give us some insights?
Yeah, sure. So thank you, Etienne, for your question. So our main way of assigning taxonomy to prokaryotic metagenomes, assembled genomes, is using a resource called GTDB, the Genome Taxonomy database, and its associated toolkit. It's by the same authors of CheckM, and that's our primary way of using genomic information to try and assign these mags in. And we've actually just got a funding to work with GTDB to actually be their source of mags, such that they can enrich the tree of life with our mags alongside the isolate genomes. So that's how we actually performed the assignment of taxonomy to our mags. So I'm trying to read the rest of the question.
It's about using chimeras and sequences, statistic tools to.
Yeah, so I think the other part. So once you've got the genome as a whole, you've got far more information. If you're looking at just individual contigs, contigs and metagenomes, there are ways of doing taxonomic assignments as there. Kraken is one example, but I think what we're actually finding now is that sour mash is becoming our go-to tool, partly because of the scale that it can operate, and also the fact that with Kraken you have all sorts of issues that as it gets more information rich in certain places, it actually becomes less precise in its taxonomic assignments because there's more k-mers in common. So actually, but sour mash has ways of overcoming this, and so actually sour mash is becoming our tool of choice.
Thanks, Rob, for those answers. Is there any burning question right now still?
Maybe we can move on.
So yeah, so we will move on and can keep the other points for the round table later on. So thanks again, Rob. And
there's another one. I'll answer that in text.
Okay, thank you.
Okay, and Damien, now if you can introduce the next speaker, thanks.